Welcome to the class 390.51, which is the introduction to our programming language for bioinformatics. So this class is given in the computer science department of the University of Crete, and this video will be a short introduction to bioinformatics for computer scientists. Okay, so what is bioinformatics? Definitely there are several definitions for the word bioinformatics, but in principle what we do every day is to use computer science tools, so software, and statistics and mathematical models to analyze biological data. So specifically for biological data, what this course will be about will be genomics. Either DNA sequences or gene expression data. So methodologically, what we do is to apply this set of tools and biological data as input in order to acquire some knowledge. For the biological system that we study. For example, we use as input gene expression data to find out which genes behave differentially depending on if we use a specific drug or not. So for example, we use data that describe the expression levels of each gene. This is this one. Then we find out that, for example, this is the outcome, that gene BXF3539 is upregulated, so it has a high expression values when a person takes drug X, and has low expression values when it does not take drug X. The analysis, so this will be the input, and here we apply statistical tools to get this outcome. Now the knowledge that we acquire here is to find out which genes are affected by the drug X. Now I use the word gene is affected implying that there is a causal relationship between the drug and the gene expression levels, which means that the drug affects the expression of the gene. However, in most of the times we will not be able to infer this causal relationship. So in general, we will have that gene expression level, or a variable a here, is correlated with the value of another variable, which is here taking or no taking drug. This will be the general statements that we will do throughout the course. And again, this is a knowledge that if you tell me if a patient gets a drug or not, then I can tell you something about the gene expression level. Now this is important biologically because it allows us, for example, to find out a set of genes. So from one gene, this gene here, we can go to a set of genes that all of them are affected by the drug, and therefore they might participate in the same biological process regulating, for example, the metabolism of the drug, or participating in a biological process that is affected by the drug, all of them. Now, why to use bioinformatics to answer such questions? The thing is that biological systems are very complex, as probably you know. So in the human genome we have something like 20 to 100 thousands, so 20,000 here to 100,000 expressed proteins that all of them, or expressed genes, the same thing. I will use the term proteins or genes interchangeably, that some of them are expressed, some of them are not expressed, some of them are affected by the lifestyle that we have, some of them are not affected by the lifestyle we have. The expression or not of this protein set here defines what we call a phenotype. So the expression of these proteins or genes together with the environment, so here environment plus the proteins or genes, defines what we call a phenotype, which is exactly how we look like. Not just the appearance, but how healthy our systems are, how strong is our metabolism, what is our weight, and so on. So lifestyle and genetic material define us. And since there are so many things that may define us, and I'm talking about the proteins of genes, which are about 20 to 100,000, then it's very difficult experimentally to test one by one or a few of them every time if they affect the phenotype or a specific phenotype. And since the technology got advanced and we were able to measure expression levels, for example, of all genes simultaneously, then we started to think, okay, how we can use all this information to get some knowledge for what we are in reality. And then we figure out that the problem is actually statistical. So in order to find out how or if a specific drug or a specific environmental condition or the age or the gender, how it affects our gene levels, then we must apply statistics in order to understand the effect of each gene of these variables on the expression level of the genes, or vice versa, the effect of the genes on these variables that we study. So we have to apply computer science methods that will handle on one side this large amount of information, and on the other hand they will provide the tools, software, to analyze in a few hours or in a limited time thousands of genes. And this is not possible to do either experimentally or by hand. We have to use software and computer science tools. But then this is not enough, because the nature of the data is often stochastic, as we will see later on, and therefore we need statistics to test hypotheses or make predictions. For example, if I will tell you here that a person has a high expression levels for genes gene X, Y, and Z, can you tell me something about his health conditions after 10 years, let's say? Inferences or predictions of this kind need statistics to be achieved. Therefore, I hope that I have convinced you that bioinformatics in reality at least methodologically is a set of tools and software and methods from computer science and mathematics and statistics on large biological data. Now this is the methodological part. However, the story does not end here. After we get the results, we must somehow interpret them. So a second big part is the interpretation of the results. Coming back to our example of gene expression levels, if I will tell you that if some people take drug X and using bioinformatics and all the statistical analysis, we figure out that gene A, B, C, and D is upregulated, so has high expression levels, while genes L, M, N have low expression levels, then what does it mean for the regulation of the expression level of these genes? What does it mean for our ability to affect the expression levels of these genes? Or what does it mean for the well-being of the person that these genes are upregulated and these genes are downregulated? Perhaps then drug X will have a side effect because these genes are downregulated or because these genes are upregulated. And all this is the interpretation of the results. And in order to do that, you need to have some biological knowledge. So with that, I want to stress that if you are a biologist and you want to do bioinformatics, then usually you have to spend a quite considerable amount of time on studying algorithms, studying statistics, studying how to write code, and some math. On the other hand, if you are a computer scientist, then you have to spend quite a considerable amount of time studying biology, genetics especially, some statistics, and some math. If you are a mathematician, then you have to learn how to write code and to learn about biology. So it's not an easy task, and actually this is the reason that it's very, very interesting. And I hope that in this semester you will, if you are a computer scientist, you will be able to deal with biological problems and biology will be interesting for you, and if you are a biologist, you will get all the knowledge needed to handle problems that are interesting for you because of your studies. Thank you very much.